Now, the immune system, or immune response rather, consists of two interconnected arms, the innate immune response, or innate immunity, and adaptive immunity. Well, innate immunity refers to that portion of the immune response which is responsible for detecting components shared by all pathogens that we have to deal with as invaders. An interesting concept in terms of how this might work, and not immediately obvious how it works, but indeed it turns out to be the case. Adaptive immunity is something different. Adaptive immunity refers to a type of immune response that's molecularly crafted to mold and detect individual antigens that are specific to individual pathogens and individual types of pathogens. These two work together in some rather interesting and mysterious ways that we'll go into later, but nevertheless it's important to keep in mind that there are these two equal and equally important aspects of the immune response, innate and adaptive immunity. Innate immunity was really first discovered by this great scientist of the past, Elie Metchnikoff, shown here working in his laboratory about 100 years ago at the Institut Pasteur in Paris. Metchnikoff's great conceptual advance was the understanding that inflammation, which is what happens after you become infected by the introduction of a bacterium or a virus. This is why your cuts become red and inflamed and hot and painful if they go untreated. That this process of inflammation is a protective process of immunity and not a manifestation of tissue destruction. In other words, as bad as it looks, this is actually a representation of how the body is responding to invading microorganisms to rid the body of those microorganisms rather than a reflection of the microorganism's capacity to mete out damage on the host's tissue. Prior to this point, this idea was completely foreign to scientists. Now, the story goes that one of the most important observations that Metchnikoff made was actually made while on vacation on the beach with his family. And like many modern-day scientists who bring their computers with them on vacation, Metchnikoff, in the age before computers, brought his microscope and studied the reaction of starfish larvae to the introduction of foreign material, such as bacteria, introduced experimentally into the larvae. What Metchnikoff found was that a series of cells would come in, surround the bacteria, and in fact quite visibly engulf these microorganisms, shown here as these red oblong creatures contained within this cell. He named these cells macrophages because they were large cells that ate bacteria, macrophage, large eater. The bacteria were taken to the interior of the cell, and Metchnikoff further found that if he stained these cells with a dye that partitioned into acidic elements within cells and other things, the bacteria stained red, further indicating to him, and in fact correctly to us, that the bacteria, after they entered into the cell, were delivered to an acidic intracellular compartment, which we now understand and know as lysosomes. Now, bear in mind, this was well over 100 years ago, and long before even membranes were truly discovered. Now, these bacteria eventually stopped growing. They died after being taken up by the cells, and eventually disappeared. And Metchnikoff further understood that as a process of degradation. So to summarize then, Metchnikoff really has to be credited with providing us a great deal of not only conceptual understanding, but in many ways even experimental detail that has defined for us the innate arm of the immune system. This is a system that recognizes shared pathogen-derived components that are recognized by toll-like receptors, as well as a variety of related receptors that we won't have a chance to talk about. It is powered to a very large extent by cells which are referred to collectively as phagocytes. And these include the macrophage, which are the large cells that Metchnikoff first identified, as well as neutrophils or granulocytes, polymorphonuclear leukocytes, which actually are smaller than macrophages, and indeed Metchnikoff had seen these too. He referred to them as microphages. These mediate protection by activating cytotoxic mechanisms, and as you've seen quite graphically, by actually physically eating, clearing, and degrading extracellular bacteria and other types of microorganisms. Now, almost at exactly the same time that Metchnikoff was working elsewhere in Europe, Another terrific scientist, Paul Ehrlich, was also hard at work, and you can tell from this image that Ehrlich was a terrific and great scientist because he has one of the hallmarks of great scientists, which is an incredibly messy office. So here he is sitting, reviewing some notes, and what Ehrlich's contribution was to show for us how the other main arm of the immune system was organized and what it did. This is called adaptive immunity. What Ehrlich identified was that individuals, whether be they humans or be they animals, that are immunized with various types of microorganisms or bacterial-derived toxins, make a response in the blood that is inherently protective. And in fact, Ehrlich coined the term antibodies to describe this response, which has the ability, obviously, of protecting us against whatever it is that had entered into our systems or into our bloodstreams. Antibodies see individual antigens that are specific to individual pathogen types. So if you immunize with one type of bacteria, you will not necessarily make antibodies that will recognize and protect a second individual against another type of bacteria. So in this way, the system is fundamentally different from the innate immune system. It's crafted in a molecular fashion to the molecular entity that happens to be coming in. The way the cellular mechanism whereby the adaptive immune system works is using not so much macrophages, as we know now, but rather making use of antigen-specific lymphocytes that indeed make these antibodies that recognize and kill the infected target cells, either separately from, or as you'll see in a minute, in conjunction with the macrophages.